see, there are big differences in the health and the size of the plants. The mycorrhizal ones are doing much better.
Well, it appears that the endomycorrhizal nature of essentially all the upland plants is an important element in the Chernobyl cesium story, at least so far as the sheep farmers are concerned. But before we go into that, it's worth pausing to look in more detail at the structure of a root and how it functions in iron uptake. And this is a model of a root tip, the part that is pushing through the soil. The surface you see here isn't root surface cells. It's actually the sheath of mucus and bacteria and debris that coats the surface of the root, the rhizosphere it's called. You can see the root cells if you look down on the cross-section. These projections out here are the root hairs, and they're extensions from just single epidermal cells, the epidermis, this outer layer of cells here. Now, ions and water can go into the root hairs or into the epidermis, and then they can cross the cortex, diffusing down an electrochemical gradient inside the continuum of living cells, the symplast. Or, instead of that, they can move not inside the cells but outside along the cell wall or apoplast route, and that's the way that calcium goes. But the apoplast route is blocked once you get to this layer here, the endodermis, because its walls have a layer of thickening, the Casparian strip, which is waterproof. So it blocks the apoplast pathway. So entry into the stele, this central part, has to be via the symplast of the endodermis. And once into the stele, the ions can diffuse across to the xylem and from there be transported up to the shoot. You can see the xylem better if we look at a longitudinal section. The xylem cells now down the middle, they're very young ones, and you can see this spiral lignified thickening in their walls. And then at the tip of the root, here's the root cap. Now this protects the root tip as it pushes its way through the soil. It's very tough going out there. I mean, it's rough. And this root cap gets worn away, and then it's replaced by cell divisions in the meristematic zone here. And then ions and water are taken in all over this root tip zone down here. Although, of course, they can only get up to the shoot if there's xylem present. But as the ions are taken in, they tend to deplete the soil immediately surrounding the root in a zone surrounding the epidermis. And for a particular ion, the width of this depletion zone, as it's called, will vary, depending on the mobility of the ions. For something that's very immobile, like phosphate, and phosphate tends to be in very short supply in the upland soils, it's an extremely narrow zone, millimetre or less. And in effect, the root's exploiting only a tiny volume of soil for phosphate. This is where the root hairs help, because they protrude out beyond this narrow band around the epidermis, and they form their own depletion zone. Effectively, they're exploiting new soil. But even so, in the phosphate-poor upland soils, there's still great difficulty in getting in sufficient phosphate to support the plant's growth. And this is where the endomycorrhizal partners are so useful. This is an endomycorrhizal hyphae, just one of them. You've got to imagine lots of these sticking out of the root. And they protrude way out into the soil, absorbing phosphate very efficiently out here, far beyond the root hairs. And then they deliver the phosphate into the cortex, and we've shown the hyphae actually ending inside the cells here with this little spiral structure. But what about the other ions? Well, some of them, like nitrate and potassium, do enter with the fungus. But more important for these other ions is the fact that with phosphate restriction on root growth removed, the whole root system grows much better. So it exploits far more soil, so it takes up far more of the other ions. And so you might conclude that endomycorrhizal partners would actually facilitate the uptake of cesium in the upland soils. That certainly seemed a logical conclusion to me.
Yes, that's exactly what we expected, but it's not what we found.
Oh.
In laboratory experiments, we found that young mycorrhizal heather plants actually absorbed less cesium than the non-mycorrhizal controls. However, although the mycorrhizal plants had absorbed less cesium overall, they had transported more of it to the shoots, which is the part that the animals, such as the sheep or grouse, grazes. So the presence of a mycorrhizal association may act as a link in the transfer of radiocesium between the soil and the grazing animal.
But I'm puzzled though, Jill. I mean, why does the Mycorrhizal Association increase the transfer of cesium to the shoot?
We simply don't know, and it has to be said that there is a great deal that we don't understand about the uptake of cesium by plants. Much more basic research is required before we get to the bottom of the problem.
So far, we've been looking at nutrient uptake and then how the ions move from the roots to the shoots. But for Chernobyl cesium, that's not the whole story. Plants around here are still, many of them, radioactive. Even though they've shed leaves and they've died down to ground level every winter, let alone being burnt in heathland fires. Now, if these were lowland plants growing on a reasonably rich soil, all those shed organs, the roots and the shoots, would have contained cesium. It would have been released during decomposition, and then a lot of it washed away, leached from the soil by rain. But it's not like that on these nutrient-poor upland soils. For one thing, these plants hold on to their nutrients very tightly. When they die down or leaves fall off, they withdraw the nutrients before this happens, taking them into the perennial parts, the woody tissues or the roots. And then they remobilize them in the spring, and in that way you get very efficient internal cycling of the nutrients. The cesium or the other nutrients that are left in the plant litter, well, they're released in the surface layers of the soil, and then they're almost immediately reabsorbed by the mat of mycorrhizal roots. So you get very tight external cycling of nutrients too, and there's very little lost by leaching. In these nutrient-poor uplands, this is particularly true for cesium. Cesium, you'll remember, is an element closely related to potassium, and potassium is in short supply in the upland soils. Work at Merlewood on the roots of a common upland grass, Agrostis capillaris, has shown that when the grass is grown in potassium-deficient soil, the root's capacity to take up cesium is up to 12 times greater than that of roots grown under optimum conditions. Which means that on the potassium-poor upland soils, the grasses on which the sheep graze have a high capacity for taking up radioactive cesium. So the problem facing the Cumbrian farmers is that because of the way this upland ecosystem works, Chernobyl cesium has been concentrated and well and truly locked up in the vegetation. So let's just recap on why those early predictions were so wrong. First of all, the acid peaty soil in upland areas means that cesium is more available to plants than in lowland areas. Secondly, potassium deficiency in upland soils, and hence upland plants, increases the uptake of the cesium. Upland plants also tend to have an efficient system of internal cycling, which means that cesium, once taken in, does not readily leave the plant. And lastly, mycorrhizal fungi cause tight external nutrient cycling, and the Merlewood research has shown that they increase cesium transport to shoots, at least in heather. If more had been known about the physiology and ecology of upland plants, then we might have been better able to predict the effects of that Chernobyl fallout, and we certainly wouldn't have misled the sheep farmers so badly. A fuller understanding of the ways in which mineral ions are taken up and recycled in different ecosystems is essential if we're to be able to predict the outcome of any future Chernobyl-like incidents. And until we have that understanding, we'd better be very cautious indeed about releasing any potentially toxic ions into the environment.